Today we will discuss about the very important topic, polymorphism of lysosomes. Lysosomes are spherical bodies so these were first observed by Christian Dewey in liver cells. These are single membrane bounded cell organelles. So they contain enzymes. So these enzymes works in acid media by using water molecules. Hence these enzymes are called as acid hydrolytic enzymes. Okay? So all the lysosomal enzymes are works in acid media by using water molecules. That means they destroy the substrates in the acid media with the help of water molecules. Hence these are called as acid hydrolytic enzymes. So these lysosomes exhibit very specific feature that is called as polymorphism. Hence lysosomes are called as polymorphic cell organelles. So these are the brief information about the lysosomes. So the lysosomes are spherical bodies. So they were first observed by Christian Dewey in liver cells. So these are single membrane bounded organelles. They contain the enzymes. So these enzymes destroys the substances in the acid media by using water molecules. Hence they are called as acid hydrolytic enzymes. Here the enzymes are considered with the digestive process. So these enzymes are called as digestive enzymes. These lysosomes exhibit a specific feature called as the polymorphism. Hence these organelles are called as polymorphic cell organelles. So let's discuss the polymorphic nature of the lysosomes. So what is polymorphism? So the appearance of more than two different forms in the life period of the organism or by the cell or by the cell organelle. Okay? So the polymorphism means occurrence of more than two different forms in the life period of an individual organism or by the cell or by the cell organelle. Here, so this type of the polymorphism we are seeing in the siphonophores or coelenterates. So the lysosome also exhibit the polymorphic nature, but this polymorphism of the lysosomes mainly observed in the process of digestion. So during the digestion process, the lysosomes lysosomes contains different substances. So based on the contents present in the lysosomes during digestion, the Roberts describe four types of lysosomes. One is primary lysosomes. Second one is secondary lysosomes. Third one is residual bodies. Fourth one is autophagic vacuoles. Okay. So based on the contents present in the lysosomes during digestion, D. Roberts described four types of lysosomes. So number one is primary lysosomes. Second one is secondary lysosomes. Third one is residual bodies, and fourth one is autophagic vacuoles. So let's we discuss the formation of these four kinds of lysosomes during the process of digestion in the cell. The first one is primary lysosomes. So the primary lysosomes are the first kind of lysosomes. These are formed or synthesized by the Golgi complex or by the endoplasmic reticulum. So the primary lysosomes are synthesized by the Golgi complex or by the rough endoplasmic reticulum. So these are the first kind of lysosomes. They contain the enzymes, and also they preserve or they store the enzymes or substances. Hence they are called as the storage granules. Another one is the secondary lysosomes. The secondary lysosomes are formed by the fusion of both primary lysosomes and phagosomes. The secondary lysosomes are formed by the fusion of both primary lysosomes and phagosomes. Okay, so here we have the primary lysosomes are formed. But what are phagosomes? So phagosomes are the vesicles. They are formed during the process of endocytosis. So what is endocytosis? So endocytosis is the process in which the particles enter from outside to the inside. Outside to the inside. Here, the food particles are foreign granules. They are absorbed into the plasma membrane. Then they are they are taken into the cytoplasm by the enfoldings of this plasma membrane. So and these are the enfoldings. The plasma membrane at these enfoldings gets pinched off into the cytoplasm in the form of a small vesicle. So the plasma membrane at this enfoldings get pinched off in the form of vesicles. Okay. So these vesicles are entered into the cytoplasm. So these vesicles are called as endosome. So these endosomes are two types. If the endosome contains solid substances, this is called as phagosome. If the endosome contains liquid substances, it is called as pinosome. So endosomes are two types, phagosome and pinosome. Okay. So let's be coming to the formation of the secondary lysosomes. After the formation of these primary lysosomes and the phagosomes, these two are unite together and to form as the secondary lysosome. So this is the secondary lysosome. Okay. So the secondary lysosome is formed by the fusion of both primary lysosome and the endosome. This secondary lysosome consists mainly two substances. So one is the enzymes. So another one is the food particle. So that is entered from outside to the inside. So this food particle is digested by this action of primary lysosomal enzymes. Okay, so the food is digested by the lysosomal enzymes. So after the digestion, the digested substances are diffused. The digested particles or substances are diffused into the cytoplasm. Hence, the secondary lysosome consists only the undigested particles. So, the secondary lysosomes with undigested waste products called as the residual bodies. So residual bodies are the third type of lysosomes. So they fill with the waste products. These are the wastes. Then these residual bodies then moves towards the plasma membrane and join with the plasma membrane. After joining with the plasma membrane, they expel the waste products to the outside by exocytosis. So another last one is autophagic vacuoles. So these are the special kind of lysosomes. So these autophagic vacuoles are formed during the starvation period. The process is called autophagy. Autophagy. So in autophagy, the lysosomal enzymes digest the intracellular organelles. In autophagy, the lysosomal enzymes digest the intracellular organelles. So during the autophagy, the cell organelle which to be digested it is enclosed by a membrane. This membrane is called as isolating membrane. So this isolating membrane is formed from the endoplasmic reticulum or by the Golgi complex. So after the completion of this isolating membrane, a vesicle is formed. So this vesicle is called as isolation body. So this isolation body fused with the primary lysosomes. To form as special type of vesicle. It is called as autophagic vesicle. Vesicle. Okay. So the autophagic vesicles are the special kind of lysosome. These were formed during the starvation period. So in this the process is called as autophagy. So in this process, the intracellular organelles are digested by the action of lysosomal enzymes. So in this process, the cell organelle which to be digested, it is enclosed by a membrane called as isolating membrane. It is derived from the endoplasmic reticulum or by the Golgi complex. So after the formation of the isolating membrane, a complete vesicle is formed. It is called as isolation body. Then it joins with the primary lysosomes and form as the autophagic vacuoles. Here we are observing this autophagic vacuoles contains the two components. One is the lysosomal enzymes and the intracellular organelles. But here the secondary lysosomes also contains the two components. In this one, lysosomal enzymes, and second one is the extracellular organelles. That is the difference between the secondary lysosomes and autophagic vacuoles. Okay. So these are the next. These autophagic vacuoles are these autophagic vacuoles, the intracellular organelles are digested by the action of lysosomal enzymes. Okay, so these are the four types of lysosomes are during the process of digestion process. Okay.